Can you see my screen? Yeah.
It's perfect. Thank you.
Okay. Thanks for the great introduction, and it's my pleasure to share our recent work with you today. And my title is gut-brain circuits for fat preference. So obesity is a growing public health problem facing the world today. So obesity prevalence has risen dramatically from the 1975 to 2016. So around 2 billion adults were overweight all over the world. And of these, over 650 million were obese. And this number is still increasing. So the main cause of the obesity is overconsumption, especially on fatty and sugary foods. For example, my favorite food here is Peking Rosé Dark, the seared, crispy breast skin dipped in sugar. And after I finished this, I ate another ice cream and a cake tart. So here comes the question: what drives our insatiable appetite for sugary and fatty foods? And what we want to do is to uncover the neural basis for sugar and fat craving, which can provide different strategies to help control the sugar and fat appetite and prevent obesity. So our previous work showed this preference paradigm. So a freely moving mouse is presented with two bottles. One bottle contains sugar, here using glucose, and the other one contains artificial sweetener. So using concentrations at which both are equally attractive. So at the beginning, the mouse drank equally from either bottle. So the bar shows the leak raster for glucose in red and the artificial sweetener in blue. So the densities are quite similar. So after it was exposed to both bottles over 24 hours, the mouse almost only drank from bottle containing glucose. And this graph plots the pre and post preference index, like for glucose, showing the dramatic increase. And importantly, this dramatic behavior switch also happens in knockout mice, lacking the sweet taste receptor. So although mice cannot taste sugar or artificial sweetener, they can still learn to recognize and choose the sugar. So it proves that sugar craving is independent of sweet taste, most likely as a result of the post-ingestive effects. So if a population of the neuron could be identified that respond selectively to the consumption of sugar, it may provide an entry to review the neural basis for sugar craving. So our two graduate students in the lab found that the neurons in the caudal nucleus of solitary tract, the CNST, are only responsive to sugar, but not artificial sweetener. And when they use the trap strategy to specifically silence these sugar-evoked neurons, the mice can no longer develop the preference for sugar when compared to the control animals. And then they monitor the real-time neural activity of the CNST neurons in brainstem, synchronized with the intestinal delivery of sugar or artificial sweetener. And they found that these CNST neurons are activated by glucose, but not by the artificial sweetener. And the bilateral transaction of the vagus nerve could abolish these responses. So the vagus nerve is a conduit for transmitting the sugar signal from the gut to the brain. To further validate the role of the vagal nerve, they performed this vagal ganglia imaging and showed that a subset of the vagal neurons are selectively activated by intestinal delivery of sugar, but not artificial sweetener. So taken together, they discovered this sugar-specific rather than sweet taste-specific gut-brain axis to create a preference for sugar and motivate consumption. Then what about fat? Another indispensable nutrient. So when the mouse is given a short access to choose between the water and artificial sweetener, sweet, of course, is very attractive to mice. They drink a lot of artificial sweetener. And then we do the same thing with fat. here we use intralipid, a very commonly used fat emulsion. So mice innately leak a lot from bottle containing fat. So this immediate attraction to sweet and fat is mediated by tongue. So to explore the development of post-ingestive fat preference, so a naive mouse is given two choices between the fat and artificial sweetener. We know that artificial sweetener do not trigger post-oral preference because it doesn't activate the gut-brain axis. So that it enables us to monitor the emergence of the fat preference. We make artificial sweetener more attractive. So at the beginning, the mouse leak a lot from the bottle containing artificial sweetener, so showing blue color. However, after 40 hours exposure, the mouse switch its preference for fat, overriding the innate attraction to sweet. So the mouse nearly exclusively drink from bottle containing the intralipid in the last 2000 leaks. And this graph shows the dramatic switch from the less preference for fat at first to unquenchable appetite for fat in the end. Importantly, this unquenchable appetite for fat is independent of taste, because in the 30 minutes immediate test compared to the wild-type mice, the taste knockout mice can no longer distinguish the fat from water, so they consume randomly from either bottle. However, when we leave them there for another 48 hours, the taste knockout mice, although blind to the taste of fat, can still develop strong post-ingestive preference for fat. To understand the neural basis for this fat preference, we first try to find the neural population in the brain that selectively activate by consumption of fat. We expose the separate cohorts of mice to fat and fat-free texture controls, and then examine their brain of CFOS as a proxy for neural activity. So we found that compared to the control, the fat can strongly activate the bilateral CNST neurons that show in these yellow boxes. And the activation of CNST by fat is dependent on the post-ingestive recognition, because when we directly infuse the fat into the gut and then do the same thing, we found that the intragastric application of fat is sufficient to activate the CNST neurons. And if this fat activates CNST neurons are essential for the development of fat preference, then blocking their function should prevent its development. Therefore, we use the trap system to target the Cre recombinase to fat activate the CNST neurons, and then bilaterally injecting the CNST with a Cre-dependent tetanus toxin construct to silence this fat activate CNST neurons. So after the tetanus toxin were expressed, these mice were tested with two-bottle preference assay. So here I showed the fraction of the artificial sweetener versus intralipid consumption after 48-hour test. So these silenced mice were unable to develop the preference for fat over artificial sweetener. So mice consume less intralipid when compared to the control. So it demonstrates that these CNST neurons are required for driving fat preference. To investigate how the fat signals transferred from the gut to this brainstem CNST neurons, we infuse the fat stimuli into the gut and simultaneously record the neural activity of the CNST neuron by fiber photometry. And this image shows where we record from and the track of fiber. We found that the CNST neurons are not only activated by direct infusion of sugar, but also by fat. So after bilateral transection of vagus nerve, these responses were completely abolished, suggesting that the vagus nerve is required for transmitting the fat signal from the gut to the brain. So if this fat detection and preference is mediated via the gut-brain axis, it should be possible to monitor the activity of the whole circuit by imaging the response of vagal neurons while delivering the different stimuli in the gut. So we implement this in vivo vagal ganglia imaging platforms. We exposed a ventral window into the ganglia, and this picture is a representative field from a vagal imaging session showing the soma of this GCAMP, this calcium indicator expressing neurons. And the two fat evoked vagal neurons are highlighted as the examples. We can see this beautiful responses locked to every stimulus delivery. Usually will repeat the same stimulus for 3 times. And the neuron is classified as a responder if it activates the responses in 2 or more trials. And this heat map shows around 150 responders activated by the fat stimulus. As I showed previously, that the intestinal application of glucose can also activate a subset of vagal neurons. So therefore, we want to know how vagal sensory neurons encode these 2 nutrient signals, fat and sugar. So we alternately administer 10 seconds glucose and 10 seconds fat into the gut 3 times. And as a heat map show, we identified 2 distinct groups of vagal neurons. One group respond to both fat and sugar, while the other respond only to fat, but not sugar. So among the 2,000 image neurons, so around 8% respond to both nutrient, 8% only respond to fat. In addition, we also observed that the subset responding to both fat and sugar is also activated by amino acid, so another essential nutrient. This suggests that the subset of vagal neurons is responsive to all 3 macronutrients, fat, sugar, and amino acid. So they're named as nutrient responders. And another subset only respond to fat, named as fat-only responders. So what about the signaling from the gut to these vagal neurons mediating fat and sugar sensing? So CCK expressing enteroendocrine cells in the epithelium of intestine have been proposed to function as a sugar preference gut sensing cells. So CCK is known as a hormone that functions as a part of the digestive system. It's released by small intestine during the digestive process. So we hypothesize that the CCK may be the signal between the gut and their partner vagal neurons. Therefore, we examine the responses of vagal neurons to this the fat, sugar, and amino acid before and after the pharmacologically inhibiting the CCK receptor signaling with the antagonist. And we found its application abolished all the fat, sugar, and amino acid responses on nutrient responders. But on fat-only responders, the fat responses remain robust and consistent. So this suggests that the nutrient sensing, but not fat-only vagal neurons, utilize the CCK signaling. So principally, CCK is known as a satiety hormone in the gut, modulate the food intake by suppressing appetite. However, the function of nutrient circuit is to promote nutrient consumption. So how CCK can function both as a satiety hormone and as a nutrient preference signal in the gut. We reason that this could be easily resolved if a genetically distinct subset of CCK receptor expressing vagal neurons mediate the nutrient preference. Therefore, we use the single-cell RNA sequencing atlas from the nodose ganglia to further characterize the subset of CCK receptor expressing neurons. So this graph plots the transcriptome of the mouse vagal neurons, and red color labels the CCK receptor expressing ones. So we identified a unique subset of the CCK receptor expressing vagal neurons marked by the uretensine 2B, this UTS2B. And we obtained this Cray line and use it to express GCAM. And we found that this UTS2B expressing vagal neurons respond to intestinal stimulation with fat, sugar, and amino acid. And we can tell from this pie charts that the UTS2B Cray mice largely enrich the nutrient responders. Next, we searched for the marker gene that can specifically label the fat-only responder. We know the fat-only responder don't use the CCK signaling, so we identified the 5 minimally overlapping candidate cluster that don't express the CCK receptor, but cover the rest of the vagal ganglia neurons. And screened by nodose imaging, we found that the TRPA1 expressing clusters respond selectively to fat, but not sugar or amino acid. Therefore, we hypothesized this that nutrient sensing vagal neurons should be required for both sugar and fat preference, while the fat-only neurons could be only required for fat, but not sugar preference. To test our hypothesis, our strategy is to genetically silence the nutrient sensing vagal neurons by bilateral injecting the nodules of UTS2B Cray mice with tetanus toxin, and then perform the 48-hour preference test. As hypothesized, after 48 hours, the mice were attracted to artificial sweetener, but not glucose or intralipid, while the control mice still go for the glucose and intralipid, which means that the nutrient sensing vagal neurons are required for the development of both fat and sugar preference. Next, we did the same thing on the TRPA1 Cray mice on this fat-only responder. After genetic silencing, these mice no longer develop the post-ingestive preference for fat stimuli, but remain their capacity to develop a preference for sugar. So by comparing the gut-to-brain pathways driving preference for fat versus sugar, we uncover the two parallel systems, one functioning as a general sensor of essential nutrient, responding to intestinal stimulation with sugar, fat, and amino acid, which is CCK dependent. And it's a subcluster of the CCK receptor expressing vagal neurons, mediating the sugar and fat preference. Whereas the other is activated only by fat, and which is independent of the CCK signaling and doesn't overlap with CCK receptor expressing vagal neurons, mediating the fat preference. So next, let's move our eyes on what happened in the gut. The food after ingestion arrives to the small intestine, where the carbohydrate and fats are broken down into the single sugar units and free fatty acids. We reason that the sugar and fat can be detected by different sensors expressed on the enteroendocrine cells. So our pharmacological experiments have previously demonstrated that the sodium glucose link transporter 1 functions as a gut receptor that recognizes the glucose. So now we extend the specificity of these findings by generating the SGLT1 knockout mice and examining their vagal responses to intestinal stimulation with sugar and fat. So these heat maps show that all the vagal responses to sugar are abolished in these mice. By contrast, the responses to fat stimuli remain unaffected. So what about the fat detection in the gut? So dietary fat, once ingested and digested, is thought to be sensed by a number of putative gut receptors, including the fatty acid translocus CD36, G protein coupled receptor GPR40 and GPR120. We anticipate that one or more of these receptors would be used to transmit the fat preference via the gut-brain axis, Therefore, we use CRISPR-Cas9 to generate the mice deficient in all combinations of CD36, GPR40, and GPR120, including all the single, double, and triple mutant. A key prediction would be that the loss of the essential receptors would abolish the vagal responses to this intestinal stimulation to fat. We express the GCAMP in these knockout mice and assess their fat and sugar responses by nodose imaging. And we found this GPR40 and GPR120 double knockout and the triple knockout mice nearly lost all the fat responses, but the sugar responses were unaffected. It demonstrates that the GPR40 and GPR120 are the essential sensors of this intestinal fat signals to the vagal neurons. So if these knockout mice can no longer detect the fat via the gut-brain axis, then they should also fail to develop a preference for fat behaviorally. So we monitor their leaking for fat versus artificial sweetener. So at first 4 hours show at the black bar, and the leak of the last 4 hours show as a red bar. And we can tell that after 48 hours, the wild-type mice are able to switch their preference for fat. And the CD36, GPR40, GPR120 single mutant, as well as CD36, GPR40, CD36, GPR120, this double mutant were same as wild-type control mice. By contrast, the GPR40 and GPR120 double knockout and the triple knockout mice were no longer capable of developing a behavior preference for fat. So taken together, sugar and fat, these two indispensable nutrients, rely on different receptors and cells, but use a dedicated gut-to-brain system to detect and report the presence of intestinal sugar and fat to the brain. We uncovered these two parallel systems, one functioning as a general sensors, driving the sugar and nutrient preference. The simple but very elegant logic of this circuit is that after the gut cells are activated, the circuit does not need to preserve the identity of the specific nutrient stimuli, and needs only to ensure that the emerging gut-brain signals triggers the behavior preference. And another one is the fat-only circuit that driving the fat preference. So it will be of great interest to determine the brain targets for sugar and fat. So how CNST broadcasts these signals to the rest of the brain, compare and contrast their functions, and will provide valuable strategy to modulate our sugar and fat craving eating habits and help to combat obesity and other metabolic diseases. And finally, I would like to express great thanks to my advisor, Charles, who supports me all the way and all the time. I would also like to extend my appreciation to my amazing teammates and friends, the Wei Yi, Catherine, Zhengyuan, Ashley, Andrew, Laura, Hao,